Translation occurs in the ribosome and is the production of a polypeptide from mRNA synthesized by transcription.
The ribosome has a small and large subunit made of RNA and proteins. Those two subunits come together in the presence of mRNA. There are 4 sites on the ribosome, 1 for mRNA bonding and 3 for tRNA bonding. Specifically, the A site holds the tRNA that carries the next amino acid for the chain. The P site holds the tRNA that is currently building onto the polypeptide chain. The E site releases tRNA molecules after they have added their amino acids to the chain.
Amino acyl tRNA synthetase links tRNA with the appropriate amino acid based on the tRNA's anticodon. ATP is converted to AMP and the tRNA becomes charged. tRNA takes the place of the AMP, breaking bonds between the phosphate groups in the process, creating AMP. The enzyme releases the charged tRNA, aka amino acyl tRNA.
tRNA consists of an anticodon, which contains 3 nitrogenous bases and an amino acid. These 2 parts are connected by another form of RNA.
The small ribosomal subunit binds to the 5' cap and scans a strand of mRNA for a start codon. Methionine is the amino acid that starts the chain because it is connected to the start codon. The start codon helps determine the reading from the segment of mRNA that is translated by a specific ribosome. When the first tRNA molecule holding methionine, the ribosomal subunits and the mRNA come together, the translation initiation complex is formed with the help of initiation factors. Energy is lost in the form of GTP.
After initiation, the elongation cycle begins. tRNA anticodons match with complementary mRNA codons in the A site. Sometimes a codon and anticodon do not have complementary third bases, but an amino acid is still added to the chain. This is called wobble. Elongation factors help lengthen the amino acid chain. GTP is converted to GDP, the process is endoergonic, and peptide bonds are formed and amino acids are linked. The entire chain is transferred from the tRNA in the P site to the tRNA in the A site. The carboxyl end of the chain attaches to the amino acid.
tRNA molecules move through the ribosome through translocation. The tRNA starts in the A site, receives the polypeptide, moves to the P site, transfers the polypeptide to the new tRNA in the A site, moves to the E site, and is released through the exit tunnel. The mRNA moves with the tRNA so that each codon is translated once and only once.
Eventually, the ribosome reaches the stop codon, which ends translation. The release factor enters the A site instead of tRNA, and the polypeptide chain is released when the bond between the tRNA and the polypeptide is hydrolyzed by the release factor in the P site.
The subunits, polypeptide, and RNA break apart from each other.
Many ribosomes can translate the same strand of mRNA at the same time. Ribosomes working on the same strand can form structures called polyribosomes.
Certain peptides dictate the polypeptide's destination. These are called signal peptides and are recognized by signal recognition particles. These particles move the whole ribosome to the ER, ensuring that the polypeptide ends up in the right place.